Welcome, everyone. Thanks again for joining the second Big Data Seminar of this academic year. This Big Data Seminar series features overviews of various contexts related to big data, offered every few months. This current seminar features machine learning and tools. Later topics will include brainwave data, CT imaging, wearables, and real-world evidence. The theories is made possible by the partnership between the CCTSI Biostatistics Epidemiology and Research Design program and CETA. If you are curious to know more about the resource and for statistical support available through CETA, I encourage you to check out our website for more information with a link that I will post in the chat. We advertise our big data seminar to the campus through emails, LinkedIn, and our Twitter account. So you're welcome to follow our social media as well to get update about our education opportunities offered by CETA. So today, our seminar speaker is Dr. Arjun Krishnan, who is an associate professor in the Department of Biomedical Informatics at CU Anschutz. Before CU, he was an assistant professor at Michigan State University. Arjun received his PhD in computational biology at Virginia Tech and conducted his postdoctoral research at Princeton University. In addition to biomedical data science research, he is passionate about open science, research training, and creating diverse and inclusive learning environments. Very interesting research interest. Thank you so much for joining us, Dr. Krishnan. And with that, I will pass the Zoom session over to you.
Thanks a lot. Hey, everyone. So just a couple of things, if there are, feel free to interrupt me during the middle of the talk in case if you have any clarifying questions. I also have the chat window open, so in case if you want to post something there, I'll look out for anything from you folks. Otherwise, I'm going to dive into this stuff and hopefully I'll get through some. What I want to do today is to talk about 3 projects that are going on in the lab, among many others, and use these projects to talk about 3 major data types that we work on, and also use this to talk about 3 ideas in machine learning that we like to always think about. All right. So before I get into the details, I want to make sure that I acknowledge this fantastic group of people that I get to work with every single day. These are awesome graduate students, postdocs, undergrads, high school students, programmers, research instructors. So we are a very diverse group in every access possible, including our backgrounds and research training. And we are working on a variety of cool problems in biomedical data science broadly. And what we think about all the time is to see, okay, how do we get to the bottom of genetic, molecular, and cellular cellular mechanisms underlying complex traits and diseases, and do this in a very general way. So we are not focused on any one disease area, but we develop, we think about broadly, how do we get to these mechanisms across diseases? And how do we do this? By using data-driven discovery, using AI and machine learning. Okay. And the key for us for data-driven discovery is to be able to use publicly available data as effectively as possible. So we work with and handle massive, diverse public data collections. So to give you a sense of the data types that we work on, for example, we work with a large amounts of bulk and single cell multiomics profiles. We work with molecular interaction networks, a lot of unstructured text-based information in terms of publications or descriptions of samples and experiments, lots of genetic data. This is individual level or population level genotype, phenotype data. And then we ground all this on a very firm, manually curated knowledge bases and ontologies. Right? So the question is, these are all very different data types, and they're also massive data types. Okay. And when I say the question is, how do you integrate them and use them effectively? So in our group, we develop algorithms, benchmarks, and software to advance every step of data-driven discovery to use public data sets like this. And so we work on a diverse types of projects. Some of them involve, how do you design better data harmonization annotation methods? Others think about data integration and creating, data representations of very high-dimensional data. Then how do you build machine learning models that can help us make predictions and interpret mechanisms at the genetic, molecular, and cellular levels? We also develop and release benchmarking frameworks, datasets, and tools so that others who are developing methods can compare their methods to ours or to anything in the field in a very on a level playing field. And the last thing is we also release a lot of open code software and web servers so that other computational scientists can use our methods, and other biomedical researchers can use our tools, but using a web interface without any coding. Okay, so I'll talk about all this in a bit down the line. Right. Okay, so before I go to the next slide, the top left of the slide has this, sense of bulk and single cell omics, right? So just to give you a sense of the scope of data that I'm talking about, what I am referring to when I say public data of a particular type is every single data point that has ever been generated for that particular type, and that is available in the public domain. So for instance, when I say, let's take one single type of data, which is gene expression data, which is our omics data, genomics data. So what I'm showing you on the table left is a consolidation of human data that's available from this many repositories, like GEO or ArrayExpress, and this data is from, these tallies are from 2020, so the numbers are much higher now. So even if you take the 14 most common experiment types, it clocks to about almost 27,000 data sets, and that's almost 1.3 million genomic profiles. And this is just genomics data. And there are many, many other types of data sets that I talked about in the previous slide. Okay. These data come from hundreds of tissues in the context of a vast variety of genetic, environmental, physical, and clinical factors. So therefore, the data is very diverse and can be really useful in many different contexts. They've been generated by scientists from across the world and deposited to public databases over the past 25 years. Okay. And now, if you include not just data from humans, but also data from many other research organisms or model species that we use constantly in biomedical research, the number is almost like 2 million, genomic profiles. And this is, again, just one data type. So this is the scale of data that I'm talking about. Okay. All right. Just to drive home this point, we are huge believers in the incredible value that existing public data can bring. So which means that instead of only generating new data to study new avenues in biomedical research, we also believe in using existing data as effectively as possible. And we and others have shown over time and time again that reanalyzing existing data can lead to generating new hypotheses to answer new questions, can check reproducibility of original findings. You can perform integrative analysis by combining many existing data sets. And you can essentially review earlier studies to see if they support any new findings that you find in your lab. And of course, they are very good at meeting urgent scientific needs because the data already exists. Okay, so what I'm going to do today is to give you a flavor of the types of projects in the space of using public data, using machine learning and AI. And the three projects, so I'm going to pick three projects, and each of these projects, I'm going to focus on three specific data types, and then which then lead to three ideas in machine learning, right? Okay. So the first one is about the fact that if you take the human genome, and if you ask how many genes in the human genome have been experimentally characterized to the level that we know what function these genes perform, which pathways and processes they participate in, and which diseases they might play a role in, that number is very small compared to all the genes in the genome. It's like close to about 5,000, 6,000 genes have some experimental characterization. The remaining genes do not have any knowledge. So what we do often is to think about this problem and use large graphs. These are large networks where nodes are genes and interactions represent physical or functional relationships. And we then develop machine learning models that learn patterns in these large graphs to identify novel roles that genes play in processes and diseases in the body. So the ideas are like this. So, like I said, most genes in most species, including humans or research organisms, are highly undercharacterized. So, what we do is to think of genes as working with each other as part of a social network inside cells in our body. Every single circle here is a gene. The connections are, relationship, functional relationships between these genes. And then what you can do is to say, okay, I am interested in learning about a particular pathway, phenotype, or a disease, and here are a bunch of genes that I already know to be associated with this disease, with this, let's say, phenotype. And here, those genes are A, B, C, D, and E. What you can do is to locate them in the network and then essentially do something very intuitive, that occurs to all of us, which is to look at the neighborhood of these genes and ask if there are genes that are very tightly connected to these known genes. So, for example, the nodes with these dotted circles are very good candidates of new genes that can participate in this original pathway or phenotype that we began this investigation with. Okay. So this idea is pretty straightforward. What we do in our lab is to do a short twist in this idea to not just look at local neighborhoods like this, but to turn the problem in another way. So think of the toy example on the left of the slide where every circle is a gene and the connection and the thickness correspond to strength of interaction. Now I tell you that here there are blue genes which are, let's say, related to a particular disorder of interest, and then the red genes are genes related to completely different disorders, okay, that are not very different from my disorder of interest. So here is the question for all of you. Okay, if I show you this, then I tell you that here is a new gene. I have no idea about its disease, role in any disease, but I tell you which other genes in the network it's connected to, and then I would like you to take a shot at telling me whether it belongs to my disease of interest, which is blue, or not, which is red. So whoever thinks it is blue, can you give me a thumbs up? Okay, Ian. Okay, so others think it's not, doesn't belong to this blue category, or you think it's belong to the red category. Okay, some of you. Okay, I'll let you folks continue thinking about this. But this gene is very likely belongs to the blue category. And the intuition is very interesting. It is not because this new gene is connected to blue genes. Sorry, new gene is connected to the blue genes, but it shares very similar network neighborhood to the blue genes. So this is like how if you log into LinkedIn, LinkedIn is going to say, here is your middle school friend who you might want to connect with. You two are not directly connected on this platform, but you two have common friends on this platform. That's why the platform gives you this suggestion, right? So it's a very similar idea. So you can turn this idea into a some machine learning framework. So to give you, how this is possible, you can do things like, okay, so let's take this example of this is an example from a real study that we did. We looked at how genes interact in specifically the human brain, and that's the network at the bottom left, which is all red edges. And then at the top, we wanted to use this network to identify novel genes associated with autism spectrum disorder. So to begin this process, again, we tried to ask, okay, what are the genes that are known to be associated with ASD? And then can you use that to find additional genes? So we collected that information, and then we had several types of genes related to ASD, some that had strong associations and some that had weak associations. So you can also incorporate this idea into a machine learning model. And I'm going to show you a very simple model and how these pieces fit in. Okay. So for example, in an equation like this is the optimization function. And then I can tell you that, for example, this Y tells you whether a particular gene is known to be associated with the ASD previously or not. The L tells you how much evidence do we have about this gene association, whether it's strong or weak. And then the X encodes what is the connection pattern of each of these genes in the brain specific gene interaction network. So once all these things are captured, the model essentially figures out what is the importance of every single gene in determining whether its neighbor is an ASD gene or not. Okay. And that's essentially very similar to the intuition that we sort of worked in our head when we were solving this toy example. Okay. So for example, for this gene being a blue gene, being connected to these three genes is very important. Therefore, they have positive values. And not being connected to these gray genes is important. Therefore, they have negative values here. So that's what is automatically captured by this model. Okay, so the problem is even harder than this. So the way we are taking this to the next level is to say, I've been talking about gene interaction networks in humans, but we have networks in many other species as well. And it's really important to be able to translate information across humans and model organisms because it's really model organisms are key in doing experimental validations or functional testing. So the question is, how do you transfer anything that you know about genes related to pathways, cell types, tissues, traits, or diseases seamlessly between humans and models? And when you have all these networks from multiple species. So the challenge is that these networks are, again, very large. So for example, if you just look at known protein-protein interactions across all these species, we have about 1.59 million edges interactions. And then if you look at functional relationships that are predicted, it is about 500 million interactions. So these networks are very, very large. So an idea that we often do is to say, okay, we don't want to work with this whole network as a single unit, but we want to create a representation of the nodes in the network in some high dimensional space. And this idea is called node embedding. This is an idea that was, first proposed in a very popular way in 2016. And we have, had several iterations of better and better methods. And the idea is the following. If you look at the network at the top left of this particular slide, the idea is to take a network like this and then create a representation like the graph on the top right, where every node is a point, and then two nodes are close to each other in this new space if they also have very similar network neighborhood in the original network. So you will see that nodes that have similar colors are close to each other because they are also in similar network neighborhoods on the left. So the way we do this is to train a neural network model. And then what the neural network model allows us to do is to capture this numerical representation of every single node, which is what is plotted on the top right, given a graph. And what we have done in our lab is to take the original node-to-vec, the algorithm is called node-to-vec, which is the most popular node embedding algorithm. We have taken that and extended it to weighted graphs. A weighted graph is when the edge also has a weight, which is a strength of connection. Okay. So we made some improvements. Then now what we do is to take the networks from multiple species and then use node-to-vec plus to create a representation where all the genes from all the species live in the same space. Now you might have seen plots like this for PCA, right? This is similar to PCA, but every dot is a gene. And then 2 points are close to each other if they have very similar network connections in any of these species. So I can even plot where the genes from different species live. They are all living in slightly overlapping but different regions in the space. And what this allows us to do is to say, now if I did something like a genome-wide association study in humans, identified a bunch of genes, I can use this space to find out what are the most functionally equivalent genes in any of the other model organisms so that I can then do experimental follow-up. The same works for the other way around as well. If somebody was working with a rat model or a C. elegans model, and then you did something like a genetic screen and you've identified a bunch of genes related to a particular phenotype, what you can then do is to use this space to ask, what are the most equivalent human genes to the genes that I identified using my genetic screen or drug screen in a model organism? So again, I'm skipping over a lot of details, but it's possible to do these things. Okay. All right. So what I told you now is to use large network graphs and machine learning to basically ascribe functions or disease associations to genes in humans and model organisms. The second idea that I'm going to talk about is this knowledge of something that I talked about earlier is that we want to use public data as effectively as possible. But using public data is not easy because of the fact that many of these data sets are described in a very poor way, and I'll talk about this. The descriptions of samples and experiments come in the form of unstructured text. And the machine learning idea that I'm going to talk about is how do you turn text into numbers so that you can use machine learning algorithms on text very easily. Okay. So here is an example of the description of a sample in the public domain. Okay. It's a block of text This is not like a nice table. And this block of text has a lot of technical jargon. So somebody needs to be an expert to basically read this and understand what is going on in the sample, where did the sample come from, what are they really studying here. Okay. So this is unstructured text with a lot of ambiguity. Now, there are also other problems with this unstructured text. One is that scientists use very different words to refer to the same thing. Somebody might say pulmonary, others might say lung. They are referring to the same thing. Somebody will say myocardial infarction, others will say heart attack. There are also cases where, for example, if you wanted to read the description and say which tissue was the sample isolated from, if you think that okay, this is just text, I'm going to do something like search, you'll get many hits for tissues in the body. For example, this was blood drawn from patients with kidney cancer and macrophages were isolated for transcriptome profiling. Once you read this, you know the data came from macrophages, but you can't do just text search to find out which tissue the sample came from, especially if you want to do this for millions of samples. The fourth problem is that many times researchers will not include every piece of information that's important for the sample. Somebody will say that we study psoriasis, but they will never mention the word skin. Or somebody will say they study cerebral palsy, but never mention the word muscle. Right? And then finally, there are many typos in this description. And then even if the description has some structured information, structure looks like this. For example, here are all the ways in which people encode age. Okay. So you have to somehow have some ways of capturing all these different ways of any attribute being described. So what we've been doing in our lab is to develop natural language based models that can take the unstructured text as it is and then make inferences about this data. So the idea that we use is called word embedding. This is a very popular and very strong approach developed in NLP and computer science many years ago, maybe 6 or 7 years ago. And the idea is very simple. It is to say you can train a neural network model that can take a large amount of text. Let's say it can be every single publication that has ever been, is out there. Runs into 28 or 30 million publications, right? And their titles and abstracts and full text articles. And then you can essentially train a machine learning model. Let me skip this part to ultimately achieve the following. Okay. What you can do is that after you train a neural network model, the model will allow you to turn every single word in the text corpus and represent it as a set of numbers. Now, two words that mean very similar things or keep co-occurring together over and over in the text corpus will end up with very similar numerical representations. So therefore, you can reverse engineer this, that if there are two words or ideas or keywords or phrases that have very similar numerical representations, they are likely to mean similar things. So you can sort of use this idea, turn this idea on its head, then use it in the following way. Words are very hard to use as input for traditional algorithms, but every traditional statistical or machine learning algorithm can take numbers as input. So therefore, turning words into numbers is very powerful. All right. So we use something called character embedding, which allows us to, embed even individual characters so that we can do things like, avoid issues about misspelling and all that stuff. But that is just a small detail. All right. So the way we use word embedding to solve the sample annotation problem is this. We went ahead and curated a large number of samples that are annotated that basically came from various tissues in the body. Okay, this is about 12,000 samples. And then what we essentially do is to turn the description of every single sample into a set of numbers. Now, the way we do this is very simple, okay, intuitively. A sample description has many words. Okay. We turn every word into a set of numbers. And then once we have the embeddings of all the words in the description, we simply average them to get an embedding for the entire sample. So this is our way of turning the description of a sample into a set of numbers that captures the description. Right. So we do this for all the samples for which we have previously annotated them to different tissues. Then what you can do is to train machine learning models to basically predict the following. Given the text-based numerical representation of a sample, can you predict whether the sample came from heart or not? And we train another model saying kidney or not, and another model saying brain or not. So you can train a model for every single tissue in the body. And once you have these models, then you can take the description of any new sample do the same pre-processing and convert them into numbers, convert the descriptions into numbers, and then feed them as input to the pre-trained models, then get predictions of whether that sample is likely came from heart, kidney, liver, and so on and so forth. And then you can use the probabilities to infer the tissue of origin. I'm hoping this makes sense. If not, please interrupt me. All right. So one major point uh, that I want to say is that I'm talking about tissue of origin here, but you can basically use this to predict any sample attribute. For example, which disease were they studying, or what experimental conditions were they using, which technology were they using for their particular experimental assay. You can turn this into any prediction of any sample attribute, and then you can repeat this whole thing again. Okay. All right. So here's a cool thing. If you look at, let's say, omics data or any other data that's collected out there, the molecular data that's being collected is so different. Some of the omics experiments are measuring gene expression, others measure proteomics, others measure metabolomics, methylation, or whatever. Comparing these molecular data to each other is very complex. However, all these samples are described by researchers using text. So therefore, you can actually train models based on text descriptions of samples from one technology and easily apply them to classify samples from any other technology. Okay, so it's very technology free because it just uses text, so it translates pretty well. So we have shown that it works well across data types as well. All right, here is the final project. Okay, so in this project, I'm going to talk about very important biomedical problem, and then for which we use gene expression data. That's the public data type. And then I'm going to talk about how do we make predictions about these samples from gene expression data, which turns out to be pretty important for solving the biomedical problem. Okay. So the problem that I'm referring to is this idea that we want to see how do we reuse hundreds of thousands of publicly available omics profiles to characterize the genetic and molecular basis of sex and age differences in physiology and disease. I think all of you will really appreciate this, that many physiological processes and many diseases and disorders show sex differences and they show age differences because many of these disorders do not manifest and occur throughout the lifespan. They occur or show up in certain age groups or at certain times along the human lifespan and seem to be quite different across males and females. So for example, if you take rheumatoid arthritis, it occurs way more frequently in women than men and in closer to 40 years of age. And then something like autism spectrum disorder, it's a developmental disorder. It seems to occur way more frequently in boys and girls and very early in lifetime. Okay. So almost every disorder is like this, but we lack our understanding of molecular mechanisms that result in age and sex differences is very poor. Okay. So we want to see how to increase our understanding of these factors and to do this using publicly available data. So our idea was this, okay. We have so much experience working with transcriptome data in the public domain, and we actually shown the following. You can actually take these large amounts of gene expression data that's out there and then build these gene interaction networks that I talked about earlier, especially interaction networks that are specific to different tissues in the body. And we can use them to make predictions about disease genes or pathway related genes in specific networks. So we have shown this over and over in many studies. So our idea for studying sex and age specificity is the following. We said, okay, instead of using all transcriptome data, we are going to take this data, separate them by sex, and separate them by age groups. Okay. Once you have separated them by sex and age groups, then we can actually build sex and age group specific gene interaction networks, and then use that to study sex and age specific disease mechanisms. Right? So the logic is pretty good, right? So we're going to separate the data and then study every single group. So that was the idea. However, we hit a major roadblock on step one, which is that most of the publicly available data has no information about sex or age. So which means that the researchers did not describe anything about the sex or age of these individuals. But the data is there. So we have the gene expression profiles, but not sex or age annotation. Okay. So this is predict pathologic state of affairs. So what we decided to do is to So remember, for all these samples, what we have is the expression profile of all the genes in the genome, right? For these hundreds of thousands of samples. So what we did is to actually start curating for the subset of samples for which this information is available. You can skip the top part here. We essentially curated about 613 studies, which are transcriptome studies, which totals to about 30,000 samples from two different technologies, microRNA-seq, and they are almost equally distributed across females and males, for which we have sex information and age information. So this is equally split across sex. And then for age, we have very good representation for almost every age group along human lifespan. Okay. So the bars here show how many samples there are at every single age group, which is along the y-axis. So you on the left you can see we have samples from almost every age group and from different technologies and from both females and males. So this is about 30,000 samples. So now what do we do with this? We can do the first obvious thing is to say, okay, can you predict the sex of every single individual from which we got every sample? This is very easy, and we are not the first people to show this. Hundreds of other studies have already shown this, that you can easily look at genes on the X and Y chromosome and make very high good predictions. So for example, exist is a single gene on the X chromosome, and if you look at the expression level of that gene, you can basically tell if it's high, it's invariably female. If it's very low, the sample is invariably from a male. Here is an opposite scenario. KDMD, KDM5D is a gene on the Y chromosome. If its expression is very high, the sample is likely to be from males. If it's very low, it's likely from females. What we are able to do is to go one step further and say, we are not just looking at genes that are sex biased, but we can do this in an age group specific way because we have this age group classification. So we ended up identifying a large number of genes that show differences between males and females, but also those that show this difference only in specific age groups and along the human lifespan. So, for example, this ANOS1 seems to have sex bias just in the adolescent age group, and we actually connected this to some biomedical mechanism that's related to adolescent-specific, disorders, which I'm not going to go into here, but you're welcome to look at our preprint. Okay, what we also want to do the other case, right? We not only want to predict sex, but we also want to predict age group. So the way we're going to do this is to take all these age samples for which we have data, which is these 30,000 samples, divide them into 11 age groups, and then do the following over and over, which is that let's take one particular age group as an example. Let's say fetus. We're going to call that the positive age group. We're going to take everything else and call it the negative age group, right? So one age group is positive, everything else is negative. Once you have this data, we can train a machine learning model. I'm going to show you a very simple version for the sake of understanding. This model will do the following. You can take every single sample and the expression profile from that sample, and you can provide that as input. X1, X2, X3, and so on is the expression level of gene 1, gene 2, gene 3, and so on. It'll take the expression levels as the input. And then what it's going to do is to, based on the expression levels of all the genes, predict whether the sample came from fetus or not. Okay. So the way you figure out how do you combine the expression levels of all the genes is by supplying many samples with annotations. Okay. So you keep showing this model samples from fetus or not from every single sex, and then the model is trained, and it essentially figures out how should I weight every single gene's expression so that I can make this prediction as accurate as possible. Okay, so I'm going to show you the performance of a model like this using a heat map. This is kind of complicated, so the best thing to do is the following. The x-axis, right, the y-axis here shows every single age group here is the model, is a machine learning model that we trained to predict that particular age group. So for example, the top one was a model that we trained to predict fetal samples separate them from non-fetal samples. Every column here corresponds to the original age group from which the sample came from. So therefore, we want the diagonal to be very dark. If the fetal sample should classify fetal samples correctly and not the other ones, right? So we want this to be dark. I'm going to call these boxes target. Okay. I'm going to call the ones right next to the diagonal as bordering, the ones that are next to that close by, and then everything else is distant. The reason I'm doing this is the following. we actually summarized all these cells in this heat map, and then we get a pattern like this, which is kind of amazing because of the following reason. Okay, the models get the most correct predictions when the samples are from the right age group. They assign the next highest predictions to samples from nearby age groups, and so on as the samples go away from the target age group. It is very interesting. This is good, right? Because a fetal sample gives highest probability to samples from the fetus. And the next highest probabilities are samples from infant. The next highest probabilities are samples from children, and so on. It somehow understands age, the relationship, chronological relationship between ages, even though we never told the model how the age groups are related to each other. Okay. So this is very satisfying for us. That's how we know the models are meaningful. And then we basically see this irrespective of which sex you're looking at and which technology you're looking at. Anyway, the final point that I want to say with this is that remember I told you the model essentially figures out how important every single gene is in making a particular prediction. So this itself is very informative and it can tell us something about the underlying biology. So this is an example where you go back and interpret the machine learning model to gain biological insights. Because what these beta 1, beta 2, beta 3 essentially tell you is the strength and direction of importance of each of these genes in classifying a sample as a fetal sample. So we're going to call this collection of betas as the sex stratified fetal gene signature. Again, so you can do this for males and females separately and for every age group along the human lifespan. All right, so that's it. So I talked about 3 different projects, okay? And each of these projects used very different data types, one large networks, one unstructured text, another one molecular profiles. And then each of them solving the questions that I posted at the top involved considerably different machine learning ideas. So hopefully they were, interesting to you. So, but I'm not stopping here. Okay, I want to take just 5 more minutes to talk about a couple of things. Okay, these are aspects that are very important when you think about training machine learning models on big data. Okay, so all of us know that these ideas sound very cool. And of course, machine learning models, if you give them data, training labels, they're going to give you predictions. But it turns out that if you actually try to do this practically, and especially if you want to build meaningful models and meaningful predictions, there are so many, many practical challenges. Okay. So I want to tell you a couple of aspects of the practical setting. So the first is that, if you have ever, read about machine learning models or used machine learning models, you know that you have to evaluate them. So the most common way to evaluate machine learning models is to take all the data for which you know the ground truth, split them into training and testing, and then do something called k-fold cross-validation. So for example, you can split your data into 3 folds, use 2 folds to train the model, make predictions on the 1 fold that you left out, and ask how good the models perform in this last fold. Right? So this is very typical. However, k-fold cross-validation will give you an overestimate of model performance. It'll give you very optimistic, numbers and tell you the model is great because there are many other confounding factors that can actually give you very good results, seemingly good results. So to avoid this, in different projects, we do slightly different ways of splitting data. So for example, in some projects, we do something called data set holdout. So imagine this. So what we do is that instead of taking all your samples and splitting them randomly across the folds, we try to remember which data set each sample came from, or which lab or which paper each sample came from. And then we split the samples by making sure that no samples that belong to the same data set are split across training and testing. So which means that you only train on samples from a specific set of data sets and then make predictions on samples that came from completely new data sets. Okay. So not that the samples are different, but even the data sets are different from training. So a good scenario to think about this is that let's say you're doing, image-based model where you're taking an X-ray image and trying to predict something from the image. It's very important to do data set holdout. For example, you might have data from 3 different hospitals. So what is really important is to not split the images randomly and then do cross-validation, but to take data from hospital 1 and 2 to train, and then make predictions on images from the 3rd hospital. If your prediction is good, that is when your model has some generalizable performance. Otherwise, you're just making predictions about, how maybe hospital effect or the doctor effect or radiologist effect. Right? Another type of validation is called temporal holdout, because machine learning is all about predicting the future. So therefore, what we do is to remember the time at which each of those discoveries were made, which form our training data. And then we take our training data and split by time. So we'll say artificially, everything before 2017 becomes training, and everything after 2017 becomes testing. So this tries to, tell us how good prediction about the future is. The last is called study bias holdout. Okay. So many times what you will see is that it's very easy to make predictions about things that are already well known. And this is a problem that many machine learning models that are published out there, face. So to really understand how good we are at making predictions about things that are less well known, we take everything that we have and then identify how many times something has been studied well, and then use the most well studied items or samples for training, the least well studied for testing, and then do this in cycles. So these are evaluation schemes. Some of the things for you to think about is what evaluation metric people use when they report it in papers, or when you do this yourself. If something like area under the rock curve is reported, it's usually terrible. It's not good enough because in many biomedical settings, the problem is highly balanced, which is that the number of negatives are way larger than the number of positives. So, for example, if you're trying to predict disease, number of people without disease is way larger than the number of people with the disease, right? So this happens very often, and in these highly imbalanced cases, the area under the rock curve is a very bad metric to use, right? Okay, so it is very popular, but it will give you an inflated sense of performance. All right. So a good alternative for you is to use something called the precision recall curve. Okay. So again, if you're interested in doing this, you can go read up what this actually means. Okay. Or even do something like prediction, accuracy at top K. So it basically says that at the end of the prediction, all I want to give is to hand over the top 10 predictions to somebody. If that is the case, you can basically directly measure how accurate my top 10 predictions are instead of doing something like some convoluted metric. All right. The last piece of evaluation is this. Whatever you're ranking, it can be genes, it can be patients, or it can be images or whatever it is. When you produce a ranking or from a machine learning model, you not only have to ask how good this ranking is, you also have to ask, can I actually get similar performance by using just totally data-free ranking? So for example, for genes, if I want to rank genes predicted to a given disease, what I need to do is to not only look at my real ranking, but also say things like, okay, if I just rank genes by gene length, do I get good performance? Okay. This is a very good baseline because if genes are long, they just have a higher probability of getting a mutation. Right? It has no relationship to the disease that I'm interested in, but it still gives me good performance. Then my model is not good enough. Right? What if if I'm, let's say, thinking about a brain disorder, I can just rank genes by the level of expression of the gene in brain. This is again a prediction that's very generic. And again, I should be able to do better than this. Or I can even rank genes by how many functions is it known to do in the body. If it performs many functions, it is likely to be a disease gene. My point is that again, whatever you're predicting, you can be predicting genes or again images or whatever, it doesn't matter. It is really important to create these baseline predictions that are totally data free and make sure that your complex machine learning model is able to do better than any of these data free predictions. All right. The final part that I'm going to say is that, as important the techniques are, it is also very important to release open code and open data sets so that other researchers can do two things. One, they should be able to reproduce the machine learning models and the analysis that we did for and if they develop new methods, they should be able to in a very open and transparent way compare it to anything that we have done. So we think about this all the time. We are very, very careful about this. So what we do is as soon as we are done writing a paper, we submit a preprint, and then we release all the data openly, both primary and intermediate results. And then we release reproducible code so that you can literally go through the GitHub repository and almost reproduce every figure in the paper. So from start to finish. And we also started developing web interfaces so that biomedical researchers can use the methods that we develop without writing code. Okay. So all these are very important. So in case there are students or trainees here, I can give you many examples where this has been very useful even for us. So for example, when we released reproducible code, there was a reviewer who looked at this code for a particular paper that we were, working with with genome biology 2 years ago, and then they spotted an error. We came back, fixed that error, and then redid all the analysis and then published the paper. Now think about this: if we didn't release the code, there is no way for the reviewer to spot this error, and so we would have just published erroneous thing, because you can't just read the method section and then to judge whether the implementation is good or not. Okay, so another thing that we do, like I told you, is that as part of the data that we generate, we release. It is not just data, but we also release all these benchmarking ground truth labels within software packages, so that it's very easy for others to reuse. So in fact, if you're working with anything related to networks, you should definitely check out this Open Biomedical Network Benchmark. This is a massive collection of biological networks and machine learning algorithms that you can use together. And then if you develop new methods, you should be benchmarking on this data set. We also release Python packages as software. This is for computational scientists who want to run this on a local machine or cluster on a large scale. And then this is an example of a web server. This is called geneplexus.net, where somebody can bring in a list of genes of interest, select the molecular interaction network, and then we will spin up a cloud instance where we train a custom machine learning model for them. So this is really different from many different databases. In a typical database, you're pulling results that already exist. Here, we are not retrieving results that exist. We train custom models, generate new results for the researcher, and then bring them back to the researcher through a web interface. They are able to see the genes that we predict, explore them in the context of the underlying network, and also we provide functionalities to even interpret the machine learning model that we trained for them in the first place. Okay. All right, so that's it. So I want to thank you all for your attention. These are the folks who did all the work. I'm just merely speaking, and the people who funded my group. They are listed at the right, and I'm happy to take questions.
Thank you so much. Very interesting research topics to explore, and very great presentation about machine learning tools as well. Thank you. Any questions from the audience?
Yes, here you have one.
Hi, Ishmael. Yes.
Thank you for the talk, Professor Arjun. I was wondering, how do you account for the temporal transitions in gene expression and different conditions to reconstruct the gene regulatory networks?
Very good. So we don't take time, we don't consider time temporal explicitly, but the way we sort of indirectly, address temporal changes and disease-specific changes is that, this might be a long answer. So what we're going to do is that the way we set up machine learning model is the following, right? So we basically don't say, let's take disease, for example. Let's say we want to build a network that's specific to a disease. We won't go to public data and only take data sets that are relevant to the disease. Instead, we will take all data and then keep that on one side. And then we'll say what is known about this disease at the molecular level, and use that to define our labels in a machine learning model, and use all data as features, and let the machine learning model itself discover which data sets are relevant to the disease or not. So this discovery is done in a data-driven way instead of us manually making annotations. So the same thing happens with time. So if we are very interested in processes that have some particular temporal pattern, we will use that to define the gold standard and not mess with the data. And the model will tell us which data sets have the right temporal pattern that reflects the gold standard. Sorry, was that helpful?
Yeah, thank you.
Okay, good.
Any other question? Okay, I have a quick question.
Sure.
Are all your machine learning tools supervised learning?
Excellent. That's a great question. Most of the time they are, but okay. Actually, that's not the case. Remember, I told you I talked about two ideas, right? So let's get somewhere close to the top. So yeah, here. So one is learning patterns in large networks. In that idea, I was saying that there is this idea called node embedding. You take a graph and then represent every node as a set of numbers. That is totally unsupervised. But after doing unsupervised learning, the next stage is supervised, which is to predict genes related to a pathway or a disease. The same with turning text to numbers. Turning text to numbers is unsupervised. But after that, you take those numerical representations as input to supervised learning algorithms. So every approach is some combination of unsupervised and supervised learning.
Yeah. So when you use the word to back for turning text into number using unsupervised, I think the word to back is an unsupervised algorithm.
Yeah.
I'm just wondering like how you control for like small typos or things.
Yeah. So that's why we use something called character embedding. Sorry. So let's see if we can get to that place. Yeah, this is not bad. Yeah, okay. So we use something called character embedding to do this. Okay. So where the neural network doesn't embed entire words, it embeds every single character. So therefore, two words that have very similar spellings will end up with very similar embeddings. So that's how we deal with typos.
Okay. And all the data you use are from like public available data set.
That's right. There is even the model, we don't train the neural network model also. Even the neural network model is publicly available.
Right, because there are so many language models in the
Yeah, exactly, right? Yeah. So we use one of these modern transformers. So that, yeah.
Yeah, but I just wonder, like you said here, you extract text from Google News plus PubMed. How do you like are the data set already contains like text in the column, or you do that in your lab?
We don't do this.
Okay.
There are already neural network models that are pre-trained on these text corpora.
Oh, interesting.
Yeah, if you're interested in specific, I am happy to send you pointers.
Yeah, thank you. Any other questions from the audience?
Can I ask a quick one?
Hey.
So I'm wondering how you deal with noisy targets in prediction. Because so right now I work with a lot of biomedical imaging, and I'm seeing people who have like trained models or like built a gold standard from radiologist reports, and then trained models to predict that prediction from the image. And the generalizability of that tends to be pretty terrible.
Yeah.
Whereas I would imagine for something like sex and age, you can probably get some pretty precise target data to work with from at least from some data sets.
Yeah. Yeah. Okay. So what is the challenge that you are specifically referring to? So what do you mean by specificity?
So I think if we say, okay, maybe there's a feature in an image and radiologists are bad at assessing it, the models that predict from their reports are worse, and then the models that predict the prediction are worse. But I can imagine that if you take a bunch of data sets that have a bunch of different styles of assessment, a bunch of different models for this, and you bring them all together, you could actually probably get something higher quality than any independent like component. And I'm wondering, is that the sort of thing that you've done, and how you would think about handling that?
Yeah, I think whatever I'm going to say is going to be super obvious to you. Okay. So in machine learning models, more data is better than more complex models. Okay. So just the fact that, if you, for example, if you do the same exact problem, but using data from one hospital, many times these models will pick up exactly some quirkiness about how these images are analyzed rather than anything in the image itself. There are images where there are hospitals where they will put a ruler right next to the image to measure something, and then the image will have the picture of the ruler or a tiny mark of a stapler that they added to the image along with the report, and then that stapler is indicates that something was followed up, and therefore it's likely to be a disease, right? So the model will figure all these things out. So I think one is definitely more diverse data from other groups, and then which is again so very obvious to you, I know. So the other thing is also model interpretation. So which means that if you go back and ask the model which parts of the image you used to make the prediction, it should at least localize to the same regions that it makes sense to a radiologist, and not some arbitrary region in this image that has nothing to do with what is being imaged. So those are the only two things that I can think of.
Okay, thank you.
Thank you so much.
Welcome.
Yeah, for the interest of time, I think we should close down now.